now. Okay, so good morning everyone. I hope that everything is okay with the streaming. According to my computer, everything is fine. So I would appreciate if you could, if anyone could send me a message whether everything is alright or not. But anyway, I'm starting the lecture. So today we will talk about the immunobiology. um, just a moment, let me find the PowerPoint file. So, let's start with the role of the immune system. So everybody knows that the role of the immune system is to defend us against various kinds of pathogenic organisms like the viruses, bacteria, fungi, and several other unicellular organisms or worms. But it is not familiar for everyone that our immune system also defends us against the formation of a cancer. Since that cancer exists and also infection exists, so it means that this defense is not always perfect. There can be also malfunctioning of the immune system, which can cause autoimmune diseases. For example, diabetes, type 1 diabetes or rheumatoid arthritis, and there are many other autoimmune diseases, many of which appears at the old age. So here we can see the subdivision of the immune system. So we can distinguish between innate and adaptive immune system, but there are also other names such as non-specific or specific immune system, or there are also many names. I don't go into details in this terminology stuff. So what is the difference between the two, and what subsystems do belong to these two arms of the immune system? It is very easy to make this distinction. Yes, I have responses, that everything is fine. Thank you. So it is easy to make this distinction. The lymphocytes belong to the adaptive or specific immune systems. The lymphocytes include the B lymphocytes or B cells in short, and T lymphocytes or T cells in short, and everything else belongs to the innate immune system. And we can distinguish between the cellular and the humoral components of both of the arms of the immune system. What is the difference? What does it mean, humoral? Humoral means that it is a secreted product. So this term is used in a much wider sense. So for example, we call the hormonal system as the humoral system, I would say, because not the hormone producing cells directly act on the site of the, where they are needed, but only the secreted products. So this is also the case for the immune system. For the innate immune system, we can say that the secreted products can be different kind of signaling molecules, which include growth factors, etc. While in the adaptive immune system, the secreted form of the of this immune system are the antibodies, because they are secreted by the B cells. To be more precise, we will see that the B cells have to differentiate into plasma cells, and the plasma cells themselves will produce the B cells. What about the cellular component? Well, actually, the cellular component for the adaptive immune systems are the T cells or T lymphocytes. Why do we cluster them to the adaptive immune system? Because they have a very high specificity, so it means that they bind to the antibody with a great variety. So it means that there is something like a key and lock relationship. And that variation is generated by a unique mechanism which we will discuss today later. So we said that the antibodies represent the humoral part of the adaptive immune system. Everybody knows why are they specific. This is because there are, a lot of different types of antibodies, and some of them, specifically recognize the antigens, which are on the surface of the pathogenic organisms or on the cancer cells, etc., etc. So what about the cellular components? This is not always clear for the students. Actually, this is called as the T cell receptors. So the T cells have receptors on their surfaces which are produced in a large variety. A single cell produces only one type of the T cell receptors, but the different T cells produce different types of them. Okay, at this lecture we will we will focus on the adaptive immune system, but we will also say some words about the non-specific immune system. Alright, so here you can see these two subdivisions again. So we call that the innate immune system is the first line of defense. So it means that they have an immediate or a very rapid response to the appearance of antigens in our body. While the adaptive immune system needs pretty much time for building up itself. And I also have to tell you that these two systems interact with each other, and we will see some base of the interactions at this lecture. And on the other hand, I would like to tell you that the adaptive immune system is the innervation of the vertebrate organisms. Vertebrates are the fishes, amphibians, reptiles, birds, and the mammals. So it means that the insects, for example, do not have adaptive immune system. They solve their immunological problems using exclusively their innate immune system. So here we can see that the immune cells, can be subdivided in terms of their origin as a myeloid cell or lymphoid cells origins. So the myeloid cells, I may will show you a picture on that, but if not, then it will be present at the supplementary material, that there is a myeloid pathway where the granulocytes and also the monocytes are produced. So the granulocytes includes the leukocytes, the neutrophils, the basophils, the eosinophils you probably learned about at the high school. And the monocytes, and interestingly at the myeloid cell lines, not only the white bloods, but also the red blood cells are produced, and also the so-called megakaryocytes, which, if disassembled, produce the blood vessels, which are not non-cellular. So, as you can see, the lymphoids, the lymphocytes are produced through the lymphoid pathway, and they include also not only the T cells and the B cells, but also the natural killer cells, which have also a crucial role in the immune defense. But the natural killer cells belong to the non-specific immune system, such as the granulocytes and the monocytes, and only the T and the B cells belong to the specific or adaptive immune system. Okay, let's say some words about the innate immune system. So we can distinguish different kind of barriers, like anatomical, humoral, and cellular barriers. The anatomical barriers can be mechanical, chemical, or biological factors. Mechanical factors are the skin. So the function of the skin is not only in the immune defense, but several other functions, of course, but this is you know, the skin cells actually defend our internal organs from the pathogen by forming an insulator barriers. Well, there are also the cilia along the upper respiratory system. The movement of the cilia, removes not only the dust particles but also the bacteria and viruses. The mucus, the mucus is actually represent something like a trap. So if a bacteria get into the mucus, liquid, I would say they are entrapped and they cannot infect. Peristaltics is also a way of, I would say, immune defense, because the peristaltics, besides facilitating the forwarding of the digestive material, I would say, the feces, they have also a function of the movement of the air, so oxygenization, which means that they produce higher oxygen, which are non-optimal for the pathogenic bacteria. Or also the flushing of the tear and the saliva means that they remove the bacteria and the viruses because of the flushing. The tear and the saliva, you will see, also represent not only an anatomical barrier, but there are also enzymes inside, for example the lysozyme enzymes, which breaks down the cell wall of the bacteria. The chemical factors are the low pH in the stomach, so there's a pH 2 about. This is not optimal for the bacteria, and also the sweat in our skin makes a pretty low pH, not as low as in the stomach, but still it is lower than pH 7. The natural flora, in more modern terms, our microbiome, also a very important, I would say, line of defense, because they keep out the pathogenic bacteria. Not in 100% efficiency, because in many cases we have infections in the gastrointestinal systems, but still. Okay, there are also humoral barriers, humoral barriers such as the complement system. I will show you soon a picture about that, but you will discuss it in more detail in biochemistry at the second year about that. Interestingly, the coagulation system is also playing a role in the immune defense. And some secreted products such as the lactoferrin, interferon, lysozyme, which I mentioned, defensin. For example, the lactoferrin plays a very interesting role. The lactoferrin recruits or binds to the ions, thereby making unavailable them these elements to the bacteria. And the bacteria needs the iron, but if it is bound to the lactoferrin, they don't have access to this. Interferon, we already talked about at the cell communication lecture. Interferon is a very important signal molecules which facilitate the cellular defense against the viruses, etc., etc. So there are also cellular barriers, including the neutrophil and the eosinophil granulocytes, the macrophages. Macrophages have two important functions. One of them is the elimination of the bacteria by phagocyting them, eating them and degrading them. And the second one, you will see later that they will degrade the bacterial cells and display bacterial antigens on their surfaces, showing it to the adaptive immune system. We will have some details later. Natural killer cells are also important, and we will see later a little bit. So here I will show you the complement system in relation to how certain viruses try to block this system. As you can see that this is a cascade with multiple elements, and they have a wide range of functions. For example, they mark the bacterial cells for the destruction, and for example the last component of them makes rupture on the bacterial cell surfaces, or not only in the bacteria but also the cancer cells, thereby evoking lysis of these cells. Okay, so inflammation is a very important step of the immune defense. Many people think that the inflammation is a bad thing and it has to be alleviated, you know. uh, it depends. So normally it is an adaptive, process like the fever. But sometimes there is an overreaction, and only in this case we have to, reduce the inflammation. Otherwise, we should let them to work, because the inflammation is actually, as I told you, an adaptive process which includes, the recruitment of the cellular elements of the immune system. For example, when you have an injury on your skin surface, the very first step which occurs is the activation of the mast cells, and the mast cells will release the so-called histamine. The histamine has two different functions. One of them is the recruitment of the, for example, of the macrophages or the T lymphocytes, and also it exerts a so-called dilation effect on the capillaries. So what does it mean, dilation effect? It means that the capillaries, as you can see here, actually contain some pores which are tiny, especially for the very large white blood cells, especially the monocytes. Dilation means that this will be enlarged. So these pores will be large enough for allowing even the macrophages to leave the bloodstream. Alright, as you can see here, what the function is of the phagocytes. So they come into the place of the injury, which always includes, bacteria, and they eat them. Alright, so, and also those phagocytes which eat too much bacteria will die, and they will be eaten by another phagocyte. So this is something like a cannibalism. Okay, so let's start discussing the adaptive immune system. So once again, there are two main classes of them, or subdivisions, maybe if you like it. So the humoral immune system and the cellular immune system. So we can say that the antibodies are against the extracellular parasites, so it remains in Hungarian, extracellular parasites such as the bacteria. The bacteria, and the predominant majority of the bacteria, are extracellular, which means that they do not enter into the cells. There are some exceptions, but I mean this is really a rare case. While the T cells are against the intracellular parasites, which are typically the viruses. So two different tasks, because the extracellular parasites is easy to recognize by the antibodies, and we will see soon how the antibodies play a role in the destroyment of the bacterial cells. But as for the viruses, when the viruses are outside of the cells, so this is the initial steps of the infections, or when the viruses are propagated within the cells and they are released from the cells, so there are some stages of the viral infections when these organisms can be found outside of the cells. Then and only then the antibodies can play a role by their elimination. But normally the viruses are within the cells. They are cellular parasites, right? So how the viruses which are hiding within the cells can be recognized, so that this is the task of the specific T cells, the so-called cytotoxic T cells, and I will tell you the details when we understand how this whole stuff works together. Okay, so we can say that the antibodies are against the extracellular parasites, plus the viruses when they are outside of the cells, while the T cells, to be more precise, the cytotoxic T cells are against the intracellular parasites. Here you can see how the antibody works. So many students think that the antibody somehow, directly kills the, the bacterial cells, but this is not the case. The antibody binds to the surface of the bacteria by recognizing specific antigens. So the antigens are recognized by the antibody, and those antigens which bind to the bacteria are actually recognized by the macrophages. The macrophages has certain type of receptors which are called as Fc receptors. The thing is that we will see that the antibodies, which is actually protein molecules, can be subdivided into two parts. So this part here, I mean, I cannot show you here. So the stem part of the antibodies are called as Fc region, while the two arms are called as FAB region. It is just, there's a specific enzyme which cut the antibodies into two parts, and the name comes from this. It doesn't matter. But the thing is that the stem-like structure of the antibodies, which is called as the Fc region, are recognized by the so-called Fc receptors on the surface of the bacteria, of the macrophages. Okay, so these are needed for engulfing, phagocyting of the bacterial cells. Right, so what is the functions of the antibody in this case? Marking the bacteria for the recognition. And in this picture it is not shown, but as you can see that the antibodies have two different arms. So in one arm they can bind to one bacteria, while other arm to another bacteria, or to two viruses, it doesn't matter. So they can make a cluster. So it means that the macrophages with a single bite can eat, I would say, multiple pathogenic organisms, including bacteria and viruses. So here you can see the lymphoid organs. So the thymus and the bone marrow are the main organs, but there are also tonsils, lymphatic nodes, lymphatic vessels, and also the spleen is very important in that. So the bone marrow has a special role for the generation and development of the blood cells, including the red blood cells, white blood cells, lymphocytes, etc., etc. We will have some words about that. So the thing is that with regard of the lymphocytes, I can tell you that they are represented in a pretty large number and a large volume, a large amount in the body. So at the same amount as same weight as our brain. So it's about 1.5 kilogram. So this is a little bit difficult, but let me introduce the so-called pathogen-associated molecular patterns, or PAMs. So these guys belong to the whole system belongs to the non-adaptive immune system, but it has a certain degree of specificity. So these include those receptors, for example, toll receptors or toll-like receptors, and there are also secreted receptors, but typically they are on the surfaces of the cells. So these receptors are represented not in a large variety, such as, for example, the T cell receptors, but they recognize those antigenic part of the bacteria which are pretty conservative. Conservative means that many related or even unrelated bacteria has a very similar, structure of their surface proteins, and this is recognized by this. Okay, and every recognition steps done by these PEM receptors or by T cell receptors, so you will see that there are also B cell receptors to induce a cell signaling cascades within the cells. So this is not a simple process, but but there's a very complex background of that. So there is an interaction between the adaptive and the innate immune system. For example, at the site of the infection, if it is a skin injury, then the so-called dendritic cells appears. The dendritic cells are very similar to the phagocytes. Actually, they are also phagocytic cells, but they have a very special role, which will be also discussed a little bit later, that they are called as a professional antigen presenting cells. They eat the bacteria and then they display some antigens of the bacterial cells on their surfaces, and they move to the lymph node and show this to the T cells and thereby activating the T cells. I will show you soon how it occurs. Here, let's see an example, for example. So this is not a identity cells but a macrophage, but the same thing happens. And let's introduce the so-called MHC system, which will be also discussed a little bit later in more detail. So the macrophage, phagocytes the bacteria for two different purposes. One of them is to destroy the bacterial cells, and the other one is to display some antigens of the bacteria on their surfaces. And this displacement occurs through the MHC system. We will see that there are two classes of them. This is the class 2 MHC system, which is utilized by only the professional antigen presenting cells. So the MHC proteins are also called in the human case as HLA. MHC is short for major histocompatibility complex, while the HLA is the human leukocyte antigens. So the point here is that these two are the same. So in the medical application, we usually refer to HLA, but when we write scientific publications, in many cases, even the Hungarian, even the human MHC is called as MHC and not HLA, but don't be confused, this is just two different names. So what happens here, that there are T cells, we will see that there are three types of them, and the helper type of T cells have a T cell receptor which recognize the antigens. So these T cells are only able to recognize the antigens if it is displayed by the MHC system. So if they are only circulating in the body, they won't be able to bind them. So what is the point here? So there are, a wide range of variety between the T cells, and every T cells, expresses different T cell receptors. Those T cells which has a T cell receptor that can recognize, that can bind to the antigen displayed by the MHC2, will bind there, and as a result they will produce cytokines, certain cytokines, especially growth factors, and thereby they will activate both the macrophages and itself to propagate. So let's only focus on the helper T cells. So every clone of the helper T cells, what is a clone? The members of the clone express exactly the same T cell receptors. Okay, there are millions of different clones, but all of the clones are represented in a very low copy numbers. So when an antigen appears, or a pathogenic organisms with antigens appears, then and only then those clones, those variants which recognize the antigens will be selectively propagated. This is called as a clonal selection. So before the antigen encountering with an antigen, every clone are represented, let's say, by 50 or members or so, but after the recognition, those one which which fit exactly to the antigens with their T cell receptors will be represented, millions of copies. So that is the very point. So here you can see this is the proliferation of the T cells, or those clones which recognize the antigens. Okay, what about the B cells? Interestingly, the B cells have a function. Everybody knows that they produce the antibodies, but they are also professional antigen presenting cells with the capability to to phagocyte the bacteria, for example. The B cells expresses the B cell receptors. I can tell you in advance that the B cell receptors has exactly the same structure of this of the extracellular part of the receptors as the antibodies which they will produce. Okay, the antibodies do not have membrane bound region but have the same. So, so a single B cell has B cell receptors, and the antibodies which will be produced later has the same structure. Okay, so let's see what happens now. So the B cell recognize the bacterial cells through their B cell receptors, which triggers the endocytosis of the bacteria. The bacteria will be degraded within the cells and will be displaced by the class 2 MHC uh, system, I would say, and that will be recognized also by the T helper cells. Okay, now not the phagocytes, not the macrophages, but the B cells, and the same thing will happen. So those T cells which were proliferated by recognizing the antigens on the surface of the of the macrophages, will be in a large number and will meet the B cells, binder and producing cytokines, to be more precise, growth factors, and which will lead to the proliferation of those B cells which has the right receptors. What is the right receptors? Which is recognized as the bacteria, the invading bacterial cells. So this is also a clonal selection. So here you can see what happens after encountering with a certain pathogens. So the first step is the clonal selection. That is, in other words, the proliferation of the B cells. And the second cell is the differentiation of the of these B cells, because the B cells are not fully differentiated. The vast majority of the B cells will differentiate into the plasma cells. As you can see, the plasma cells are larger than the B cells, and the reason for that is that they produce a very large meshwork of the rough endoplasmic reticulum. So if you remember, of the function of this is the produce of secreted proteins. So actually the plasma cells became an antibody producing factory, right? I can tell you that there's also another name, immunoglobulins. And when I ask the question what the difference is between the antibodies and the immunoglobulins, students usually do not know. I can tell you the answer. There's no differences. the two terms refer to the same things. Okay, so in a much smaller number, not only plasma cells but the so-called memory cells are produced. Now this is also the case for the T cells. So the T cells also produce memory cells which can be stored up to lifelong in the body. So why is it important? This is important because building up of the specific immune system, requires days or even weeks, I would say. But if you have a memory cell with a specific recognition capability for a certain bacteria or viruses, then this time will be shortened. And that is the very point that you can have a much quick response to the pathogens. The major problem of the immune system is the time. If it is reduced by forming of the memory cells, then you have a much higher chance for a successful defense against the against the pathogenic organisms. And this is the role of the vaccination. So using vaccination, you actually mimic the pathogens, and if you are vaccinated, then you form memory cells, and therefore, you will be defended. So vaccination and memory cells can defend us against, most of the pathogens, but for example, it is difficult to say by the HIV, because the HIV itself, attack or are propagated within the helper T cells, which are part of the specific immune system. So actually, the HIV, the virus of the H, kills the specific immune systems. Okay, let's go further. Here we can see what the other type of the T cells do. These are the cytotoxic T cells. The cytotoxic T cells are actually antiviral or anti-cancer, has an antiviral, anti-cancer activity. If we consider viral infected cells, then what happens? So we mentioned that the professional antigen presenting cells display the class 2 MHC on their surfaces, okay? But actually, the class 1 MHC is not restricted to the antigen presenting cells, but they are expressed in every cell type, okay? And normally they exhibit or display the self antigens, and the immune system comes around and check whether everything is in order, and it checks that it's the self, it is not a problem. But when a cell is infected by a virus, then not only the self antigens but also the foreign antigens, the non-self antigens will be presented, the viral antigens, and then here comes the so-called cytotoxic T cells into the picture, because the cytotoxic T cells, approaches the infected cells, not only the infected cells but every cells, because it is unknown, from a large distance it is unknown which cell is infected and which one is not infected. So many cytotoxic T cells approaches every kind of cells, and those T cells which recognize the antigens on the surface of the infected cells by their T cell receptors, will form a stable connections, and let's see what okay, and two things will happen. If this connection is established, then there will be a clonal selection or selective proliferation of those cytotoxic T cells which recognize the antigens, and on the other hand, it will destroy the viral infected cells. How? There are actually two mechanisms, and one of them is illustrated here, that the cytotoxic T cells releases the so-called perforin, The perforin is an interesting structure because it is actually a tube-like structure. So there are globular proteins which are organized into the tube-like structure, and it has a pore in the middle, and through the pore the T cell will release a certain serine protease into the target, the virally infected cells. And that protease molecules will activate the apoptotic pathways of the infected cells. So it means, in other words, that the T cells will force the infected cells to commit a suicide. Okay, that's the point here. So here we can see the development of the T and the B cells. So every blood cells, including the lymphocytes, started their life within the bone marrow, while the B cells remains within the bone marrow for the further differentiation. Therefore, we call them B cells. B stands for bone marrow, while the T cells, after a certain step of differentiation, will migrate to the thymocytes, thymus in other words, and they will complete the differentiation in the thymus. Thymus T, therefore we call them as a T cell. Here comes the name. All right. So here you can see the clonal selection in other words. So here, as you can see, there is multiple different types of B cells. This is also true for the T cells. So here three variants are illustrated, the alpha, beta and gamma, but actually there are millions of different variants. So those variants which recognize an antigen, as you can see here, will be selectively proliferated, and that mechanism is called as clonal selection. After the clonal selection, there is a differentiation step again, that the B cells differentiate into plasma cells to a larger extent, and to a smaller extent to memory cells. This is also the case for the T cells. We call the differentiated T cells as the effector T cells, and the other cells will be also the memory T cells. Okay. So here we can see what the antigens are. Well, the antigens are typically parts of the pathogenic organisms, such as a virus or the bacteria. And the antigen itself, it's a complex structure, and it is composed of epitopes. The epitopes are those parts of the antigens which are recognized by the antibodies. Okay, the antibodies, like uneven surfaces, and so the even surfaces are very difficult to recognize by the antibodies. So the antigen binding sites are called epitopes. Let me introduce another term, which is the immunodominant epitopes. Immunodominant epitopes are those epitopes on an antigen which attracts the higher number of the antibodies, or in other words, which evoke the greatest immune response. So here we can also see that after binding to an antigen, both the B cells and the T cells, are differentiated to effector cells and memory cells. The effector cells for the B cells are called as plasma cells, while the effector cells of the T cells are called T effector cells. So there's no different names. So after the first exposure to the antigen, a large number of clonal selection occurs. So certain B cells and T cells clones are proliferated, but after the second exposure of the antigens, even much larger memory cells and effector cells are produced. What we can see here is what I have mentioned, that there is a relationship between the B cell receptor and the antibodies. So in a particular B cell, uh, the B cell receptor, the structure of the extracellular space of the B cell receptor is exactly the same as the antibody, which will be expressed later. Okay? The antibodies do not have a membrane-bound domain, that's why it can be released. So therefore it is possible that the B cell recognizes the antibody, they will be clonally selected and they secrete the antibodies. And the point here is that since the antibodies have the same structure, the same regions which recognize the bacterial cells or the viruses, that there is a link between them. So the triggering of the replication, the clonal selection by certain antigens will lead those antibodies which will recognize the same antigens. So that's the point here. Here you can see the structure of the antibodies. Actually, the antibodies are composed of 4 protein subunits. There are 2 heavy chains and 2 light chains. Both of the light and the heavy chains contains a variable region and a constant region, and the heavy and the variable chains differ in the length of the constant regions. They have the same size of the variable region. The variable regions are important for the recognition of the antigens. Okay, here we can see how the antibody antigens interact with each other. One thing here is that 2 antigens we can be recognized by a single antibody. The other thing is that having 2 arms by the antibodies makes it possible that multiple antigens will be clustered at the same site, and this makes the work, the job for the phagocytes easier, because a single bite will lead to the phagocytosis of multiple different, no, not necessarily different, multiple antigens. All right. Right. Okay, that's enough. So these are the 5 classes of the antibodies or immunoglobulins against the IgG, IgM, IgA, IgD, and IgE. So as you can see, the 2 of them has a non-monomeric structure. The IgM, when it is released, has a pentameric structure, which means that 5 different antibodies will bind together, and the IgA has a dimer structure. 2 antibodies bind to each other. The IgG, IgD, and IgE has only a monomeric structure. So here you can see the development of the B cells. Actually, the point here is that before encountering with an antigens, the B cells produce an IgM. There is also an IgD, but the important thing is the IgM, and I will explain it soon why it has a relevance here. So after binding to antigens, again the selective proliferation, clonal selection occurs on the one hand, and on the other hand there will be a differentiation into plasma cells and memory cells, and then the plasma cells will produce the antibodies. Okay, so alright, so here, okay, let's skip this picture, it doesn't matter. So here you have a list which enroll the differences between the 5 classes of the antibodies. We don't go into details at this moment. What can we see here? We can see here again that both of the light and the heavy chains have variable and constant region, and the constant region is longer in the heavy chains. And here comes an important statement, that the different classes of the antibodies there are made according to the type of the constant region. There's alpha, beta, epsilon, gamma, or mu, which differ to the classes. So the alpha stands for the IgA. Ig is immunoglobulin or antibodies. IgA, the delta IgD, epsilon IgE, gamma IgG, the mu IgM. That's the logic of the terminology. So we distinguish so-called hypervariable regions within the variable regions, which means that they, especially these regions, differ between the different antibodies. And they play a crucial role in the recognition of the antigens. And here you can see again that the antibodies or immunoglobulins are composed of different domains. And as we can see, also the T-cell receptors and even the MHC are composed of very similar domains. So all of these, immune molecules derives from the same common ancestors, I would say. So these domains are repeated in the in the light chains. As you can see, there are two domains, one for the variable and one for the constant regions, while in the heavy chain, the constant region is composed of three different domains. All right. Okay, here comes the point, the immunogenetic part. So how this large variety of the antibodies and also the T-cell receptors are produced. I will exemplify, I will demonstrate the antibodies, and the same logic can be applied to the generation of the variations within the T-cell receptors, with some exceptions which I will mention. So we would think that this, million or 10 million fold variation can be generated by a large number of genes. But actually, as you know, we only have about 20,000 protein coding genes, so there's a limitation. But the logic is not that. So this large variety is produced by a special mechanism, by a unique mechanism, which is only occurs with the antibodies and the T-cell receptors within the body, I would say, or within the genome, and it is based on genetic recombination. But actually, genetic recombination occurs throughout or during the meiosis. You know that. But in the in the adult body or in the body of the fetus, etc., etc., there's no changes of the genetic material except the antibody genes and the T-cell receptor genes. They undergo genetic recombination, after the conception. Right. And we will see how. So first of all, we have only three genes. One heavy chain gene and two light chain gene. They are called as alpha or kappa light chain genes. So actually, logically, two genes would be enough, but for any reason we have two light chain genes. It increases the variation, but not significantly. The evolution is not predictable in 100%, I would say. This happens. This happens. Two genes would be enough, one heavy and one light chains, but we have one heavy and two light chain genes. And here you can see where they are located on the distinct chromosomes. So the heavy chains are located at the terminal region of the chromosome 14, while the other two, the light chains, are on the chromosome 22 and chromosome 2. So what happens? And what is the logic for this stuff? I will explain this logic, and then we will finish this lecture and the rest of the material should I recommend you to work on this on your own. But this is an important stuff, and I would like you to understand the very basic stuff. So, what is the logic? So the thing is that here you can see the light chain genes. So in the light chain genes, the constant region is represented only in a single unvariable part. But there's a variable region and there's a joint region which are between the constant and the variable region, okay? And as you can see, there are different, I would say, domain or something like that. Okay, so within the V regions there are V1, V2, V3, V4, V22 and V40. So many. And there are some joint region, joint region. This is J1, J2, J0, etc., etc. So the point here is that genetic recombination, make a combination. So first one of the V region is selected, and this is combined to one of the J region, and then this will, will link to the constant region, and that is the point. So for example here, there is a selection between the V3 region and the J3 region. It's a little bit misleading because for the V3 there can be J4 or J5, so the 3 is not a, it's just a coincidence, I would say. So one of the V domain is matched to one of the J domain, and that makes, a single variant. But a problem already has remained, that there are some J region and also some V region which is unneeded, and once again, so there are some J region which are unneeded and they are removed by splicing. So the combinatorics, the combination of the V and the J region occurs through genetic recombination at the level of the DNA, while the removal of the unneeded J region occurs at the RNA level through splicing. Okay, and as you can see, here is one variant, V3, J3 and C region. So you can say that this is not a large variance, right? But I can tell you, that when we select, for example, the V3 region, then it is not a single point. So the V3 region itself is a pretty long, peptide sequence. So it means that you can select at the beginning of the V3, it's a little bit, downstream or the middle or at the end, etc., etc. So there are multiple recombination points. And I mentioned here an interesting thing, that this whole region, including the V domains, J domains, C domains, are continuous, protein coding regions. So if the recombination producing the frame shift mutation, hopefully you remember what it is, then there is a problem. So what about the frame shift mutation? There are 3 frame shifts, frame 1, 2 and 3. So actually, if the recombination is random, and it is random, then 2 of the 3 possibilities are nonsense, because they produce out of frame, variants which has a non-functional structure. And this is the case, that 2/3 of the produced antibodies has a malfunction, I would say. It's a bad protein structure. They are removed. So there is a pretty large redundancy in the production of the antibodies. Therefore, so let's see the generation of the heavy chains. It is a more complex. So one thing is that in addition to the V and the J domains, they also have T regions, as you can see here. And then now the task is that a specific V domain has to match the specific D domain, which has to match the specific J domain, much larger variety. Okay, and as you can also see at this picture, that there is not only a single type of the constant region, but 5 different types. Actually, this is a little bit a reduction because, for example, from the gamma, there are 4, gamma 1, gamma 2, gamma 3, gamma 4, etc., etc. So there are more than here, but still there are different constant domains which has to be selected, and that selection will make, you know, the different classes of the antibodies or families, the IgM, IgG, etc., etc. But don't rush. Let's go further again. So here you can see that we select one of the V region, one of the D region, one of the J region, and combine it one of the C region. So that genetic recombination occurs prior to being infected by a pathogens. So we do have these variations for both of the heavy chains and the light chains, and also for the T cell receptor genes. Okay, we have this variation before the encountering with a pathogen. So after, in those B cells which recognize the antigens, 2 additional processes will occur, will be commenced. The T cell receptors will not change after the binding to antigens, but the B cells will change. How? I can tell you in advance, but I also will show a picture on that. That one of them will be the class switching. So here you can see that the closest constant region domains are the C mu, and this is the reason why the why the B cell receptors are always IgM, because the C mu is the closest constant region, and this occurs before the encountering of the antigens. So after binding to the antigens, there will be a recombination which can, you know, remove certain segments here, so there can be IgD. If a larger segment is removed, IgG, but I will show a picture on you soon. Okay, so here, so this is how the recombination occurs. The recombination is carried out by 2 reg enzymes. The reg enzymes are interesting because they are because the reg enzymes are derived from DNA transposons. So actually the DNA transposons are, you know, intergenomic passages, I would say, but our genomes utilize some of them, and the reg generation of the reg enzymes is one of the form of the utilization. So the reg enzymes, you know, reg protein recognize, you know, these domains here, and the signal sequences are recognized, and, you know, here you can see that these 2 regions, the V1 and the J1, they always use the same number. I don't know why, because it is misleading. It can be V2 and J5, etc., etc. And then the recombination will occur, so there will be a removal of the V and the J region, and then there will be an end joining enzymes which will, you know, put together the you know, free ends. So the reg enzymes are the recombination enzyme, and there's also another enzyme which, you know, actually has something like a ligase function. Okay. So okay, so what happens here is that there's also an additional way of increasing of the variation. One of them is that there is a choice whether to use the maternal or the paternal copy of both of the light chains or the height chain genes. Okay, um, there is a constraint. If one copy of the light chain is used from the paternal genes, then the other copy of the light chains is also from the paternal. The same rules applies for the high for the heavy chains, right? So if one of the heavy chain is from the maternal allele, then also the other form is from the maternal. But there can be there can be a variation. If the light chain is paternal, then the heavy chains can be both maternal and paternal. So this is one, whether the paternal or maternal copy is utilized. And in the light chains, as you already know, there are two variants. One of them is the kappa and the lambda, so there can be also a choice whether to use the kappa or the lambda. Okay? The kappa from the paternal or maternal, the lambda for the paternal or maternal, or the heavy chain from the paternal or maternal. So these are also increases the variance of the light chain. Okay, so I said that two events occurs after recognizing of antigens in those B cells which recognize them, but only in those cells. Okay, clonal selection occurs and differentiation into the plasma cells and memory cells, but something else happens. One of them is the class switching. Okay, let's go to the class switching first and then come back. So as you can see here, this is the constant regions of the heavy chain. The light chain has only a single type of constant region. So what happens here, there is a class switching based on the recombination utilizing the so-called ACE enzyme. Doesn't matter. So it means that if there's no recombination, then IgM is produced. If the mu region is removed, then IgD will be produced. If a larger segment is removed here, the mu and the delta, then IgG, etc., etc. If the largest deletion will occur through the recombination, then we obtain the IgA, because the C alpha will be at the closest proximity to the constant region. So that is the point here. This is the class switching, which occurs only in those B cells or plasma cells, I would say, which already recognize an antigen. So this is the tiny fractions of the actual variations of this. Now, okay, this is the other process. This is the so-called somatic hypermutation. So, where the somatic means that this is not the germ line cells or germ cells, but the somatic cells, it doesn't matter. The hypermutation is the point. So, at the first, round of selection, so those B cells are selected which has a specificity to the antigens. But that specificity is not perfect. Therefore, an additional step occurs. This is the hypermutation. Additional step means that those B cells which already were selected by the antigens undergo a mutational step. So here you can see what about this, that there is an AID enzyme which converts which converts the C letters to U. So two things can happen here. One thing is that the U is recognized as T, but actually the U is an RNA base, so usually it doesn't happen, but it can happen. Or in other cases that one of the copies, will have a normal C letters, it's not a problem, but it is recognized as T, and therefore in the complementary strand an A letter will be produced. So the G is replaced here to A. Or the C letters is not replaced to U, but eliminated. So it means that here any kind of basis can be replaced. So mutation. This is a somatic hypermutation, because at the so-called hyper variable region of the antibodies, there's a large number of such kind of mutation. The mutation is blind, which means that the mutation is random. So therefore, the already specific, specific binding of the antibodies can be, the specificity can be not only, be higher, but can be also, looser or worse because of the randomness. But there's also a second selection round or steps, which means that those plasma cells which have the highest specificity to the antibodies, to be more precise, the antibodies will be selected against. So the results of the somatic hypermutation will be the generation of a larger array of the variants, which will include the higher and lesser specificity of the antibodies, and those plasma cells which produce the higher specific antibodies will be selected against. So that is the point here. So here we can see this summary table, and this will be the last picture for today, about the generation of the antibody diversification, I would say. So one of them, the basic mechanism is the combinatorial joining of the gene sequence. So the V7 binds to J2, etc., etc. This is one thing. The other thing is that the recombination points within a certain domains, can happen, at any case. So when we say that V7, it includes, a lot of different points where the recombination can occur. Combinatory joining of the light and the heavy chains mean that we select from the paternal and maternal allelic variants, and also the lambda and the kappa light chain variants. So all of this, happens prior to encountering with the antibodies. So we have all of the antibodies which are produced, or actually at this stage there's no antibody but B cell receptors. So antibodies are only produced when the specific B cells bind to an antigen. Okay, so this green color illustrates those events which happens with the B cell receptors before encountering of antigens. Two other events occurs in only those B cells which binds to an antigens, which are the somatic hypermutations, of which the point is to increase the specificity to the antigens, and also the class switching, which means that one of the Ig immunoglobulin variants will produce IgE, IgD, etc., etc. And this is because the different kind of antibodies, I didn't show the details here, but you can find it in the supplementary material. Different kind of antibodies have different kind of function. They're even located at different site of the body. The IgG, for example, are produced in a very large number, and they are present in the blood. But the IgA, it is a secreted stuff. So double check this in the supplementary material. So I think that everything was okay with the streaming, and then I would like to thank of your attention, and I will stop the lecture. Thank you very much.